Alrighty. Welcome back, everyone. Got some people trickling in, and I hope you continue to do so over the next few minutes. Um, I'm excited to welcome you back for the third session of Lab to Land Advancing Water and Soil Research for Agricultural Resilience today. We're on day 2 where we're talking all about soil. We started the day with a discussion on restoring forest gardens, had a panel on farmer-led research for climate adaptation and mitigation, and now we're rounding out the day with a great presentation and discussion on bringing soil biology testing on farm and some of the expectations we might have around improving soil biological health through management. So for this session, we've just got a poll up to ask one last time what hat you're wearing as you join this session. We're really interested to see, what's of interest to you folks, and this is the kind of data that helps us tailor our sessions to make sure they're what you want to see in the future. Perfect. So we'll just close that. Awesome. All right, without further ado, I'd just like to get set a bit of context for our session today, which is really that, as we get a deeper understanding of the importance of soil health, what that means across different ecosystems, but particularly for climate resilience, a lot of researchers are pushing the boundaries to better understand the role of soil microbes and soil biology in agriculture and sustainable agriculture. So in this session, Miranda is one of such experts and will be discussing how bringing that testing on farm and some of those methods can really support our pursuit of, climate adaptive and resilient agriculture. So Miranda Hart is a professor in biology at UBC Okanagan, and her research is focused on the microbial ecology of soil microbes and the role and applications of microbes in sustainable agriculture, viticulture, and ecological restoration. In addition to her research, she is the director of the plant growth facility at UBCO and is co-director of the new Wine Research Center, which is putting out some fascinating new research. So I'm really excited to welcome you, Miranda, and can't wait to hear what you have to share with us today.
Thank you so much, Asha. I'm really happy to be speaking to this crowd in particular because over the last few years, I've become increasingly aware of the sheer number of tests for soil biology and consequently the number of recommendations for soil biology that are out there on the market. And I feel like growers are inundated. Maybe correct me if I'm wrong when we get to the discussion, but people I talk to get a lot of information about things they should be doing to improve their soil. And a lot of it doesn't necessarily fit in with what science is saying right now. So, little preface, I'm not here to disabuse any company, consultant, soil testing, nothing. I think it's all good and it's all useful. I just want to give you insight into some thoughts I have about some of the tests and some of the amendments. There's a lot of good work out there, so I'm not going to single out anyone or anything. I'll just give you some information so that you guys can make your own decisions. And with that caveat, I will start my my slideshow. Hold on 1 minute. Hold on, it was there. Here it is. Okay. Sorry. Okay.
Looks good.
Yes. So my title is a little bit different than the manual or the agenda. I'm going to talk about testing and amendments and kind of give you an idea of what to choose if you're making those kind of choices. I don't think I have to explain to anyone here how important soil biology is for soil health. I mean, the soil food web keeps everything moving, everything working, and it is, from my opinion, as a soil biologist, the most important part of soil health. I think we can all agree that that's enough said. What we don't understand though is how. I mean, how and why and when and where. We know it's important, but we don't know what parts are important when and how. And there are so many parts, so many parts in the soil food web. Which ones do we need to pay attention to? Which ones are important in certain situations? which ones are good for disease suppression, which ones are good for nutrient cycling. I mean, we have an idea, but what we don't really know is how to link function and soil community identity, right? So this is kind of hallmark to everything I'm going to say out there. We know it's super important. We know there's a lot going on, but we're still figuring out how it fits together. I can say one thing equivocally, and I rarely say things equivocally because I feel like there's so much we don't know. Increased soil biodiversity is increased soil health. And this has been shown so many times for so many markers of soil health. I feel really confident saying we know this, right? Soil, here are just some of the things you want your soils to be doing on your farm. And we know that when you increase soil biodiversity, whatever that means, you get increased functioning along these metrics. And I will talk about more of these later as we go through the tests and the amendments, but just want to put that out there. This is the fundamental truth that I know to be true. Okay, so let's get into what you guys are experiencing in terms of options. How can you guys improve your soil biodiversity? How can you manipulate soil biodiversity? Well, first of all, let's start with the tests and then we'll move on to amendments. How can you test for soil biodiversity? Number one test I see being marketed to growers is tests for soil community analysis. And if you sent your soil in for one of these, you would probably pay around $100 per sample, and you'd probably get something like this back. A bunch of bar graphs telling you roughly what bacteria and or fungi or both are in your soils. If you're really lucky, your report might include something like this, that it actually includes certain groups. It drills down into the groups a little bit more and shows you the abundance of those. I don't know how many people get this kind of analysis, but either case, what we know about getting these types of analysis, and just to reiterate, all this is telling you is what DNA sequences have been found in your soil. Doesn't tell you who's active, doesn't even tell you who's alive, doesn't tell you how it changes, and most importantly, well, let's just reiterate so we know diversity good, and we know that some groups are bad in high abundance, right? So when we get a thing like this, we might see that it's never going to show you pathogens because it doesn't go into that much detail. It might show you a group that pathogens belong to that's elevated. But, oh, sorry. This should say what we don't know is we don't know which community is optimal for your farm, and we don't know how to change this. And we don't really know what to make of this. Even as someone who studies this is all I study, community change in soils and the effect of human land use on these communities. I can't tell you what community is a good community for your soil. I can't tell you which groups you should be more interested in than others because, like I said, you're not getting enough resolution from these tests to see where your pathogens are or necessarily where your beneficials are. The only way I can think of this being useful to a farmer, and I'll come straight out. I don't think this is useful to a farmer, is if you did multiple tests over time. But even that's not going to be really useful because you don't know what your target is. And also, the amount of heterogeneity in your soil. If you had a 100-meter plot, I would suggest you take, I don't know, dozens of samples in order to capture what's really going on in that plot, which, as you can see, if each sample is $100, it's quickly becoming incredibly cost prohibitive. So that's the first test, community analysis. I'm not going to go into detail of how you do it or anything. I'm just going to say what it is and what I think it can give you and what it can't give you. The next step that I see, or the next test that I see marketed a lot to growers is the fungal bacteria ratio. And the theory behind this is if you look at the history of this metric, there's research showing that as ecosystems advance in succession, fungal biomass becomes more dominant. So the idea is that the more disturbed a landscape is, i.e. an agricultural landscape, the higher the bacterial biomass will be compared to the fungal biomass. And over time, you get more fungi or fungal biomass. And I think this is pretty robust and true, but it's been used pretty frequently as a metric of soil health. And well, what do we know? What does it? So let's say you've had this out in your soil. What does it tell you? Well, we know that the fungal bacterial biomass ratio is primarily driven by ecosystem types. Some ecosystems, forests, have a lot more fungi than bacteria. Some systems, like grasslands, tend to have more bacteria. We don't really know why, but that's what the research shows. Bacterial biomass in general has a lower carbon to nitrogen ratio. So if you think about it in that way, if you have more bacteria in your soil, you might have less carbon compared to a soil that was dominant in fungi. Maybe. Another thing that we know is that bacteria may prefer to decompose easily available substrates, simple substrates, compared to fungi. This is true some of the time, but it's not true all the time. I think this is, for me, this is all I can say about what this ratio tells you. Here's what we don't know about this ratio. We don't know really what's driving the fungal bacteria ratio in different systems other than ecosystem type. pH is important. Land use can be important, but it's not the biggest driver. Fundamentally, though, we don't understand how this ratio affects your soil processes. We don't know how it affects soil nutrient cycling. We don't know how it affects soil stability. We don't know what ratio is optimum for your system. So like the community data, you can get a number, but you don't know what it means. You don't know what to compare it to. And yeah, you can, I think a lot of farmers are encouraged to increase the fungal biomass ratio, so more fungi, but it's not in all systems. That's not optimum in all systems. And we don't really know if that's going to result in any kind of improved functional aspect of your soil. So should you get it? I wouldn't get it if I was a farmer. I wish I was a farmer, but okay. Third test I want to talk about. So we talked about communities, we talked about fungal biomass ratio. The third thing is counts of specific organisms, groups that live in your soil. And here's just a list of just from a consultant who does this kind of tests and the kind of things that they are able to measure, right? Total bacteria, biomass, total fungal biomass, protozoa counts. You know, you have 3 different kinds of protozoa. You got flagellates, amoeba, ciliates, and you've got all the nematodes that they're counting. Okay, so should you get these done? Well, again, let's start with what we know. Diversity is good. I told you that. So that should include all of these critters. We also know that some groups are not good in high abundance. And I've mentioned this briefly before, but for example, plant feeding nematodes. You probably don't want a lot of plant feeding nematodes in your soil, right? I mean, that goes without saying. What don't we know about these lists? Again, we don't know, with the exception of actual pathogens. We don't know how these numbers affect soil processes, right? So you get a total bacteria count or a total amoeba count or a total cilia count. Will more be more functional? I don't know. And I think it really depends on your system and your ecosystem and your climate. We don't know what options are optimal for your system. So those are the tests that I wanted to focus on, and feel free to ask me about more tests later on. If I know anything about them, I can try to answer your questions, but I want to go into amendments. And this is primarily where my research lies. I study biological amendments to soil. This is a huge industry and it's growing so fast. This is a huge underestimate, $5.2 billion. And this is just the US in 2031 because this is only including phosphate solubilizing bacteria and rhizobia. It doesn't include any of the fungal or any of the other microbial consortia. So this is a fraction of how big the industry is. And there are so many products on the market. It's nuts. So here's what we know about biofertilizers and biostimulants, which are products you put in the soil to stimulate the bacterial and fungal communities. Well, this is from my research, but also beyond my research. We know that biofertilizers can work really well, and this tends to happen in greenhouse conditions in artificial soil. They have inconsistent and very unpredictable results in the field. Sometimes they work, sometimes they don't. And when I say they work, sometimes they establish, and sometimes they don't establish. Even when they establish, there's often no measurable effect in fields. And that makes sense. Fields are really chaotic, diverse systems, right? With a lot of microbial action going on. We do know they could move very quickly from where you put them into your soil, wherever the heck else they want to go. So if you're using them on your farm, they're not going to stay on your farm if they establish. They can establish on your farm and then move wherever the wind blows them, or the water takes them, or the animals carry them. What we don't know about these products is we don't know what they're doing to soil communities. There is evidence from my lab and beyond that sometimes these products, the biofertilizers, really suppress soil biodiversity, which, if you remember from the beginning of this talk, is the most important thing. Sometimes they do that. We don't know why and when. And that's what my research program is trying to figure out. Consequently, if they're changing the microbes below ground, how are they changing the plants above ground? Soils are not reductionist systems. They are really complex systems. So you can't make it better by adding one single organism. There's hundreds of thousands, if not millions of organisms in your farm. So adding just one to it, I almost think if it does have an effect, that's a problem. But that is just me thinking off the top of my head. Worst case scenario, if you have a biofertilizer and it works and it replaces what's already there, you might end up with a monoculture below ground and above ground. Best case scenario, of course, is you want diversity above and below ground. And I know, well, I'll talk about diversity above ground. I've been focusing on below ground, but you really can't separate them in my mind. Okay, humic acids. This is another product that I see being marketed to growers all the time. And humic acid, great. So many of them in soils, many different kinds. They do all sorts of wonderful things for plants and soil ecosystems. They're essential. And there's nothing bad about them except for should you be buying them? They're incredibly expensive. And should you be putting them on your farm? Again, I think I forgot to do our what we know and what we don't know for this one. What we know is that in natural ecosystems, they're really, really important. What we don't know if applying them is important or useful or beneficial. The research just hasn't been done yet. They're really expensive though, and not only financially, but at an ecosystem level cost, because most of them come from harvesting peat bogs. So do you really want to go to such an intensive way of managing your soils? I wouldn't, but like I said, the research is out on humic acids as an amendment, not as a part of the soil ecosystem. I'm just putting this one in here because nanobubbles. I heard about nanobubbles and it's a way of getting teeny tiny little nanobubbles into water that you use for irrigation. And it's used in greenhouses, but it's also used in the field. You can see on half of the screen, they've got these special irrigation devices. And it lowers surface tension, and I don't know what else it does, but it is, well, we know water is important. Are nanobubbles important? Who knows? Who knows? There's some evidence that it's really useful and important, but it's so expensive and so complicated, and we don't really know who needs this. So I don't know how many of you are considering nanobubbles, but I would hold on to your money for a little while because there's really, especially in the field situation, there's just, yeah, just wait. Just hold tight for a little while. Okay, so what should you do with this information? Well, I'll tell you, I have my own recommendation, and if you've heard me talk before, you've probably heard me say this before. The solution is so simple. It's so simple. It's just plants. Plants can do everything that those products can do. All of the things we talked about, fungal diversity, biodiversity, bacterial diversity, bacterial biomass, pollination, reduced herbivory, increased humic acid, increased soil carbon, aeration, soil structure, all of these things plants were engineered to do. They evolved with soil, so they've been doing this for 400 million years. They know how to do this, and they do it in such an elegant and sustainable way. This is the easy solution for you. So how do you maximize these benefits, right? It's a cropping system. It's not like a wild grassland. I get it. Um, you you guys, the horticulturalists, the wine growers are so lucky because you have this huge canvas in which to plant plants other than your crop. And this isn't a cover crop talk, so I'm not going to go into it, but you can manipulate the diversity of your plants. You can choose plants with desired traits, right? N fixers or brassicas for pathogen suppression. But you should also think about the adaptation to the climate and system, right? Are these plants going to do well here? Are you going to have to micromanage them? Because you don't really want to micromanage your plants. You want a plant community that's going to come in and do all these soil services for you without you having to intervene. That's the goal. So bring it all home. Sustainable farms need sustainable practices. The key to soil health, in my opinion, is diverse plant communities. So should you get testing? I mean, I haven't found a soil biological test that makes sense to me other than nematodes. I think nematodes, if you've got plant pathogenic nematodes, I think that's worth knowing. I think if you've got, serious disease issues, you can have more targeted tests. I don't think any farmer needs to know what their soil community is because I can't even interpret it, and I've been doing this for decades. Yeah, I want to thank all of my grad students who have done all of the work that I talked about. Actually, I didn't talk about that much, but all of my collaborators at the Summerland Research and Development Center and the wineries that I've worked with over the years and my funding agencies. And yeah, I'd love to chat about things that you guys are actually experiencing.
Amazing. Thank you so much, Miranda. I've seen a few of your talks now, and any question that I have that comes up regarding soil biological health, you can give such a definitive answer, whether it's we know or we don't know. So I loved how you structured that. And all right, so before we actually jump into the Q&A, I'm just going to describe a little bit about some options for the structure of the remainder of the session for our participants. So one option is you can stay in this main room and participate in a discussion. Feel free to unmute yourself and ask questions. This can get nice and lively with all of us here. Or alternatively, if you are really interested in kind of the networking option of this session, I am going to open a couple of breakout rooms with a couple of kind of general themes. If you'd like to sort yourselves into those rooms and meet some people, that's totally okay too. And we also have a room where La Vessie Chile, ACARN's manager, will be hanging out for a bit in case you'd like to connect with ACARN. So I'm just going to open all of those rooms up. Again, no obligation to join, but if that is what you'd like to be here for, feel free to head over there. But other than that, I'm going to stay here and get into this discussion with Miranda and all of you. And I'm just going to change my view so I can see all of you in a gallery. Feel free to open up your camera if you'd like all of us to see you, but no pressure there. Perfect. Okay, so Miranda, I'm going to start us off. There's a couple of questions in the chat that came up through your talk. And I guess one of the questions, I don't know if you come across this in your research or in your academic travels, but there's a comment that there's a local municipality working on a wastewater plan, and there's pressure from higher levels of government to include reclaimed water for use on farmland. And this person is wondering if you have come across any peer-reviewed data showing what maybe happens to soil biology when reclaimed water is used for irrigation in food crops.
I don't know. I mean, this is wastewater. It's not my field, so I haven't actually looked into this field. but you know, every time you water, you're inoculating your whatever is in the water. I would imagine that if they're, I mean, this is a big problem with the PFAs, right, in agriculture right now. It's in the water, these forever chemicals. They've been in the water for decades, and we've just figured it out. And people have been putting them on their fields and irrigating with them, and they're showing up in their crops. So I'm hoping that this reclaimed water has at least undergone some testing to make sure there's like not high levels of E. coli. That would be bad. But yeah, I don't know. I'm sure there has been work on this more through the environmental microbiology though.
Yeah, for sure. Again, I think this is one area where, there's lots more to know. And in BC, we have such a wonderful community of agricultural researchers, but we are kind of small but mighty. So this is maybe an example where we may look to colleagues elsewhere to
There's
find out
actually
what other
there
people
are,
are doing.
yeah, there are colleagues at my institution who do this.
Yeah,
So
perfect.
I can set you up if you want to know more.
Absolutely. We'd love at ACARN, we would love to know, and trying to keep track of everyone is definitely a job in itself, and we always love to know.
I Can you guys hear me?
Yeah,
I
Betty.
can put a comment in that because I'm in Cranbrook here, and the wastewater that is reclaimed, it goes through a treatment facility, and it only works really well for our area because we use more gravity for it to lead to a community pasture,
Mhm.
and it goes through the pivots and it goes through the ground on that, so no animal or food is directly
Gotcha.
consuming that. It's going through the soil to filter out any last bits. So
Perfect.
that might be something to look into as to what Cranbrook does and how and all the testing that they do.
Absolutely. That's a great comment. And I think like opens the question around, what crops are you irrigating with reclaimed water? That's really going to depend.
Yeah,
What outcomes you're going to have if you're doing vegetable crops. It's much different than if you're using soil to kind of filter out things for something like a forage crop.
exactly. Yeah, and that's where it's been working with the community pasture because it's just the grasslands and such. It's not a direct consumption from any person individually, basically.
Yeah, for sure. Thanks for that context. Nice to hear about projects like this happening around the province. Um, Perfect. And I think I've got a question here, Miranda, that I think you may have an answer for. So someone said that a farmer recently showed them test results that included measures of good versus bad fungi based on hyphal diameter. This person is a
Wow.
PhD mycologist, but they've been out of academia for a while. They'd never heard or read that the diameter of hyphae would discriminate between pathogens or nutrient cyclers or beneficial fungi. And they're wondering if there's any recent research about this or if you have comments.
Oh, I have comments.
Please go ahead.
That's just that's outrageous. That is, it just seems so simple, overly simplistic, and to the point of being nonsensical. No, that is like no, in no way is that a meaningful statement. So
Excellent.
yeah.
This is the Ask an Expert sessions that I love the most when we can debunk things that, don't pay money for and, that's really just nothing useful for your management. Awesome. All right. Okay, we have a question here about biochar. And I don't know if maybe, this is a big kind of question for a lot of growers, is this is another marketed product. And I wonder if in your research or again in your travels, if you've been able to look at relationships between biochar application and I guess soil biological health. We know this is good for kind of holding on to nutrients and increasing the cation exchange capacity, but is there any evidence around impacts to soil biology?
Yeah, I mean, I feel like this has been researched pretty well. In some ways, yeah, it's biochar is generally good for soil, except for if you've got really low nutrient soil, then it can actually bind available nutrients and take it away from the plants. So that's not what you want. Um, I guess with anything, it's harder to say though what if it makes economic sense, right? And you're adding, I would rather add plants than biochar, right? I don't know. I feel like the things that biochar is doing, you could recreate with just your plant community if, if you have the right conditions. But I don't know that anyone has, the cost benefit analysis isn't clear to me for biochar, although I don't think it's a bad thing to use.
Yeah, for sure. You know, lots of considerations when you're adding something to the system, and it's all very context specific. Perfect. I guess I see we've got lots of activity in the chat here. So if I don't get to your question, I'll maybe get to it later or we'll answer it after. But there was a question around, you know, when we're thinking about integrated crop livestock systems, if there's any evidence in terms of soil biological health, like we know that there's benefits in terms of biodiversity, but are there any specifics that you would consider?
No specifics, but in general, you know, you're going to have, if you have animals, you have different things going into the soil, right? And we know, at least from grazing systems, there is a really tight relationship between the animals and the soil microbes themselves. They co-adapt with or they co-evolve with each other, they're co-adapted, and they are distinct, right? Grazers have distinct microbiomes. But if you're just using them in your farming system, I mean, you're just getting different kinds of inputs, which I think is going to lead to different kinds of microbes, which might not be a bad thing.
Yeah, for sure. I think Dieter made a great comment on this around a lot of soil biological tests. They'll, you know, give you some data that, you know, we don't have any context for or really any way to actually interpret, but what they will also tell you is, you know, recommendations for, you know, regenerative agricultural practices or cover cropping or increasing biodiversity. And I think crop livestock systems can maybe fit into that as well of, you know, you're getting these kind of general recommendations that are available for free from your local agrologist that are coming with these kind of soil tests.
Exactly. Yeah, the recommendations are they can't be specific because there's no way to be specific.
Yeah, absolutely. This is an interesting one here. Someone is wondering if you've come across the work of John Kempf in the US. He's interviewed researchers and farmers on the benefits of using SAP analysis to assess success of improving soil microbiome and mineral availability. He does sell some products and inoculants grounded in evidence, but if you do know this person, do you feel it's too good to be true? It does sound pretty convincing.
So he does plant nutrient analysis by looking at
SAP.
that, whatever that is. I mean, yeah, what is that?
I guess, you know, SAP analysis, I think we have to ask the question of what crops are we looking at? Are we looking at perennials? Are we
Yeah,
analyzing
yeah.
like, the, you know, xylem and phloem and seeing what's in there and equating that to some kind of microbiological community? I definitely have some questions around this one, but I'd be curious to go take a look and maybe follow up on what your thoughts are afterwards.
Yeah, I don't know anything about this, but I know I think it's a method of measuring plant nutrient status. I don't know that it would be better than what is traditionally used, and it might be worse. I guess I'd have to look into it more.
Yeah, for sure. We'll certainly follow up on this one. And then another question regarding, I guess, types of soil tests. What are your thoughts regarding the Haney test for soil health?
The Haney test, I think that looks at soil respiration as a measure of activity, and then it looks at some nutrients, right?
I believe
Do
so,
I have that
yes.
right?
Or
Okay,
this
again,
bigger, it looks also at different extractable species like carbon species, cold water and hot water extractable carbon species through actually
okay.
sorted by their diameter. That's roughly, and yeah, respiration. So it's very, very carbon focused. The
Yeah.
so-called, I've been asked that question once in a while. I have my own opinion, but
I'm curious what it is.
yeah, I can forward you that. And while I have my own opinion, I don't want to, you're the expert, so, but
I'm
I can,
not the expert on this, Dieter, so please go ahead.
no, I will send you then the link to, I have my own opinion, but well, thanks considering that. I will send you the link maybe when you have time.
Okay.
Shoot me a mail back. Thanks.
Right, thanks. And Dieter, I'm actually going to loop back to one of your previous comments. You know, I think you've given some pretty solid advice here, Miranda, but what do you think that is some actionable advice we can give to growers based on the biological diversity tests? Like most give kind of trivial, universal advice to use those practices, but in terms of, I guess, your research and when you're looking at, you know, impact of amendments on things like what can we interpret from a diversity test?
Nothing. Nothing. Absolutely nothing. So I don't waste your money.
Perfect. Sounds
Thank you,
good.
Miranda. Thank you.
Excellent. And I guess that brings me, if I can selfishly ask one of my own questions here around, your current research and what you are looking at. There's definitely, it sounds like some validation of things that you're doing, but in terms of where you're at, kind of at the forefront of this, microbial research, what questions are you asking, and, what answers are you kind of looking for in terms of soil biological health in agriculture?
Yeah, I have kind of two things I'm looking at. Number one, I'm trying to identify the context under which biofertilizers work. So I do think that there is application for them. There might be a role for them in, field cropping systems, or horticulture, but we don't know what it is yet. So I'm testing different management practices and biofertilizers. So that's one line of work. And the other line of work is understanding the effect on soil biodiversity of these products. When they get into the soil ecosystem, how do they change it in terms of the structure and functioning? And when they get into natural communities, what do they do there? So those are my two big lines of research.
Excellent. There's, so many questions. I think, I guess the proliferation of all these products and things really points to a lot of interest in soil biological health. And I know you're at the forefront of this, but again, this type of research, like I can't even fathom in terms of these like population dynamics and sizes. Like it really is the utmost complexity. So this research, takes time, and the things that we're learning, like we can learn one thing and then have 100 other questions come out of that. So I think that's good to keep in mind when we're thinking about soil biology. Awesome. Okay, I had a feeling we'd get this question come up. So this is from Raylani, that who's a kind of a consultant working with lots of growers, and she sees the work of a specific, I guess, soil health proponent come up in like the soil food web. So Raylani's wondering if you're familiar with this specific person's work. And I know there's consultants like in the province who are offering services in terms of soil biological health. And, when we ask about the validity or if there's any like useful management interpretations, my understanding is that this particular group is looking at, that list that you put up around like various concentrations of fungi.
Yeah, no, I know,
protozoa.
I know her work very well, and I know what she does, and I think her training is great. She knows exactly what she's doing. I think she's got really good training. Like I've seen her training. I haven't taken any of the training modules, but they look really solid and really good. Um, here's the thing about soil biologists. We are so convinced that we know how important soils are, right? And so we want to share this with the world. And we want to show them that the tests, I guess my reservation is the tests, or for the reasons I talked about today, the tests she's offering, or her approach offers, they're solid. They tell us a lot about soil biology. They're good tests, but I don't know how farmers can use them.
Yeah.
That's my big problem.
Absolutely. I would agree. And I stand behind you on like the robustness of her trainings. I think they're like an excellent resource to get into soil biology and to get into the practice of identifying species under a microscope. Like that is all very robust. But it's when we're translating, that into management, like you can learn how to get a really accurate picture of kind of the diversity of a community, but when we infer that to management, as you pointed out, there's just so much we don't know yet about how this influences soil processes.
Yeah.
Awesome. Perfect. And I see a question from Tom here that I hoped we'd get to as well, that I'll just preface by saying, one thing that maybe a misunderstanding that I have come across is around inoculation of seed versus biological amendments, and some misunderstanding between the differences of those. And then Tom's question is asking about looking at mycorrhizal inoculants and their potential to kind of alter native soil communities. So maybe a two-pronged question. You can take which one you like.
Okay, so I'll start with your first one is so what you're talking about is the mode of inoculation, right? Whether or not it's seed
Yes,
coating or whether or not you're putting it in the fertigation or you're putting it in
exactly.
directly in the soil. Yeah, it same. It's all the same, right? I know these big companies, especially for field crops, have been developing seed coatings. It's just an efficient way to inoculate systems. It's all the same. I don't know. I don't think there's a big difference. In terms of mycorrhizal, so mycorrhizal biofertilizers is what I specialize in. So I do work with them, not exclusively, but almost exclusively. And yeah, these are the ones that we're showing do invade. They do move around. They do suppress soil biodiversity, which in the link between plant biodiversity and mycorrhizal fungal biodiversity is really strong. So if they get either these products, I guess what's important, I do want to make a distinction. This is an important distinction. There are products that are made in the lab that are selected for, really rapid growth, high reproduction, not necessarily for what they're going to do to your crop system, but they're made to establish and grow really fast. That makes them kind of dangerous as potential invasive species, right? And this is true for mycorrhizas. This is true for bacteria. This is true for anything that's produced. There are some producers, and I have scientists and friends who produce natural inoculum from ecosystems. And the evidence shows that these are more effective because they're matching the ecosystem with the inoculants or the species. It's in some cases, like there's big companies that are harvesting soil, like Funga, I think is the name of it. They're harvesting soil from Appalachian forests to produce to sell to forestry companies. Is that a sustainable practice? I don't think it is. But I do think that there's a difference between collecting microbes from your system, increasing them by whatever means because you think you're low. Are you low? Do you know you're low? Do you even need them? This is my big beef with mycorrhizal inoculums. Or inoculants, you probably don't. If you have plants growing on your site and you're not using fungicide, you probably will never ever need them. So if you are low, let's say you had a huge oil spill, or I don't know, you're growing in a gravel pit or something really extreme where you have no mycorrhizal fungi, then you would want to go take some from local populations around there that grow there, that are going to be compatible with the conditions, and grow them up before adding them. So those do work, and those work. It's used widely in restoration ecology. It's a lot of work. If you don't need it, you don't want to do that work.
Yeah, absolutely. And this kind of feeds us into this, I guess, clarifying statement around, compost teas and extracts and inoculants, like all of these things that, take a lot of time to prepare in a farm context. They're not credited to have like long-term beneficial impacts, or is the impact not well measured?
The thing about compost tea, I would love to do some research on these because, what you're doing is you're adding nutrients, you're adding a whole bunch of stuff, and you're adding microbes, and this is all good stuff. I think there's nothing wrong with compost tea. It's a ton of work though, so it goes back to the question: are you just adding the nutrients? Is that what your system needs, and is that the most efficient way to add nutrients? And maybe it is. But is there any kind of bigger benefit, right, of inoculating with a compost tea? And we just don't know that yet. We don't know.
Yeah, absolutely. So I think it's perhaps the latter has the impact. We don't totally know yet. But in terms of, delivery of nutrients, it's like it's going to be work no matter how you're going to fertilize. So I guess pick your poison around bringing in compost or making it or making compost tea, etc. Perfect. And I guess I had a question because you did bring up restoration, Miranda, and we see, I think in I guess even just the media and particularly in this part of the world where we have like major forests and forest remediation and mine remediation and lots of natural resource projects that need to be remediated. And I just wonder if we could think about, in terms of what we see around fungal biodiversity and soil biodiversity, like being really important to remediate systems. I wonder if you think that there's maybe been a jump, like one step too far over into the agricultural space where, returning an ecosystem to its natural state is not the same. We can't necessarily use the same principles to manage an agricultural ecosystem, which is not a natural ecosystem. So yeah, I just wondered if you had any comments around the distinction between soil biological health in the context of a natural ecosystem, one that maybe we're remediating, versus one we're managing for a specific outcome.
I mean, that's such an important distinction. And I think, number one, remember that the industry is not driven, it's driven by capitalism, right? So if it works in one industry, then let's try to get it into another industry. But then this is part of my research program. I told you earlier that inoculants often fail, they don't even establish. And especially for mycorrhizal fungal inoculants, most of them can't handle tillage. They certainly can't handle herbicide. They definitely can't handle high phosphorus systems. So if you're adding biologicals to a system that is intensively managed, or maybe it's not intensively managed, it just doesn't have the conditions compatible with their life cycle, there's just so of course they're going to fail. And I think that's what I'm trying to figure out. Okay, what practices will support the beneficial aspects of biostimulants and allow them, or biofertilizers and allow them to grow, and which ones will just cut it down. So yes, there's this conflict between adding them to agricultural systems when we know they're just going to die. But again, that requires farmers then to reapply them every year. So I don't know.
Absolutely. Good comment. I see in the chat, we have someone calling out my language, and I appreciate that. Humans are natural. I should get away from natural versus unnatural ecosystems. But when I say agriculture as a managed ecosystem, I just mean that, humans participating in that is arguably completely natural, but we are trying to manage for a specific outcome in the soil that is different than that soil would be, for example, in the Okanagan, if we're not trying to grow grapes in it. Perfect. And we've got another, I see First Light Farm. Thanks for being here. I'm glad that we've got some producers in the room. But Miranda, if you have any comments around the, I guess, kind of commonly advertised practice of adding sugars like molasses to the soil to boost activity for the soil organisms. And we often hear about things like an early spring wake up if you're trying to move things along, and adding sugars like in a compost tea, for example, if you have any comments on that.
Yeah, sure do. Who are you waking up with that? That's my number one. Who is waking up? You're waking up something that can digest glucose and fructose and sucrose. And what are they going to do for your plants, or what are they going to do for soil? Probably not very much. So yeah, you're going to get a lot of increased activity, but it's going to be from a very select part of the community. And so I don't know that I would do that.
Yeah, good comment. And it is something, like, with me as a background in market vegetable production, this is something that I've come across so many times. But it really, if you kind of think one step deeper, like with this goal of increasing soil health that we have, the culmination of all of this science, as Miranda so eloquently put, is, plants are evolved to do this. So let's just facilitate that magic to kind of happen and see what comes out of it. Awesome. Perfect. So we do have time for a couple more questions. So I'm just going to encourage, if anyone feels like they'd like to unmute and chime in, please feel free. Otherwise, I will selfishly ask a few other questions, but I see perfect. Larry, go ahead. See, we've got a raised hand.
Now I'm unmuted.
You are good to go.
Good to go. Okay, well, I sat here for a long time. Wonderful talk. As you know, the province of British Columbia is basically one vast monoculture of forests. Long term, what could you see done to increase the biodiversity of our plantations?
Are you talking forestry plantations?
Forestry plantations, yeah, most of the province is planted to, in our country, I'm in the central interior, and it's mostly one big vast pine plantation where we've eliminated all the competition to the trees. Long term, I think we're going to have to think about something different.
Yeah, I feel like those are historical, right? I know that foresters aren't doing that anymore, as far as I can tell, because it's so they're fire risks. They're they're bad news on so many levels. But I was under the impression that those were just kind of a historical approach to forestry. I hope.
Not really. No, when we took out originally, the old growth forest had a mix. It had a mix of aspen. It would have open little meadows. It would have open spots where there was grasslands. Now it's all been planted to trees.
Yeah, that's too bad.
So what I'm thinking about, because I'm hoping my great grandchildren will be harvesting some of our trees, and but it is a monoculture. What could be the next rotation to actually enhance the soil biology?
I don't know. Honestly, I don't know. For those coniferous forests are a bit different because the mycorrhizal associations they have are less. They're very, very important, but they're less specific than like grasslands or mixed systems. Yeah, but diversity is key. Diversity above ground begets diversity below ground and vice versa. So whatever comes next has to focus on that.
Thank you.
Yeah, Larry, I'm not sure if you're familiar with Dr. Suzanne Simard's research, and she's definitely hard to get on the phone, so to speak, but I would say if you haven't checked that out, there's a lot of great research from her around, fungal associations and biodiversity and how, the understory, how we can kind of restore these complex forest systems. So we're happy to put that in our resource package at the end of this if you're interested.
Thank you.
You bet. Perfect. All right. I think we'll take one more question that maybe is a nice place to end off in the plant realm, as we've discussed, is like our takeaway from this session. And Jennifer is asking, if we think about weeds, which, has a lot of connotations depending on who you talk to, but can we make use of quote unquote weeds as nature's free cover crop?
I mean, I think a lot of people are, right? And if you mean weeds, you mean just let whatever grow is growing. The problem with weeds, I don't really think about weeds, but if I had to define weeds, I would call them the invasive plants that grow in disturbed areas.
Yeah.
And so you're going to get kind of a limited range of ecosystem services from plants that all kind of grow the same way. So yeah, you can for sure, but I don't think you'll get a huge diversity and functionality if you just rely on weeds.
Yeah, great answer. And I guess I can add in terms of weeds, really got to think about our goals in terms of management here. There's definitely research that asks the question, is it actually better for carbon sequestration if we just use weeds as a cover crop rather than, need to till and get a managed cover crop established? But I think, timescales are important here too. If you want to make use of weeds, maybe you want to do it intentionally and, mow before a particular species goes to seed. So you're not building up a seed bank that's like really not diverse. So I won't get into too much about, cover cropping and weed seed banks, but we certainly have some resources from ACARN around cover cropping that we're happy to share as well. Perfect. I think that'll be where we end off. And Miranda, I'd just like to say a very huge thank you for your wonderful presentation today and astute and thoughtful questions. I certainly enjoyed our discussion, and I hope those in the room took a lot away from this. And I am just going to throw it back to Dr. Lavesicili, the ACARN manager, as we wrap up the 2025 ACARN workshop, Lab to Land.
Brilliant. I'm just sitting here basking in the sunshine. So apologies if that if I can't be seen very well. Thank you, Miranda. That yeah, soil biology has always been something that's very, very interesting to me. My background is in compost, and so learning, like understanding the different diverse populations in compost then led me to try to understand the diverse populations in soil. So yeah, I definitely learned a lot from the conversation today. Yeah, and thank you everybody for participating in the lab to land workshop today, or over the last 2 days. We have had a really invigorating conversation across a lot of different water issues yesterday, and this morning we heard about place-based innovation, integrating indigenous knowledge, and then moving forward into the session afterwards, we started to think a little bit about not just engaging with indigenous communities, but the communities at large. Like, how can we start to drive farmer-led research, and really what are the best strategies to incorporate the farmer in the research that's being done on the farm. And now moving into this final session, we're now talking about communities, but microbial communities. How can we make sure that we're amplifying, or what are the best ways to even conceptualize the interactions of microbes, and how microbes and the plants, and how we can start to support and elevate those different communities in a way that's beneficial for both us, as we're wanting to, create, develop productive farms, as well as the ecosystems as a whole. So thank you so much for participating in this conversation today. Just as a reminder, all of the session recordings will be available through the BC ACARN website, and also will be hosted on the BC ACARN YouTube page. You can expect an email in your inboxes sometime next week with all of the links to the different sessions. And we've also dropped in the chat, if you are interested to learn more about soil topics in BC, you can also check out our last year's recordings from the 2024 provincial workshop. Also, if you're eager, then you don't want to wait for this year's recordings, you can get started with last year's recordings as well. I also want to highlight a couple of our upcoming events. Starting on February 4th and running for 3 weeks, we're going to have our Seeding Success Enhancing Farmer-Centered Science Communication sessions. So this is targeted at graduate students, academic researchers, and industry professionals, extension professionals who are interested in learning more about knowledge mobilization and KTT. We've got 3 great sessions and 3 great speakers. You'll also hear again from Jesse McDonald.
You're just signing off.
Who was chatting with us this morning. And yeah, so please feel free to connect with ACARN. You can go through this QR code if you'd like, or you can just send us an email at info@acarn.ca. We'd love to hear more from you. And in that same vein, we really love your feedback. We would love to hear how these last 2 days were for you, so we can continue to develop and create really engaging and enriching events for the agricultural sector in BC. So Asha is going to drop that link in the chat box, and you'll also see it pop up when you close this Zoom session as well. But with that, I would just like to say thank you once again, and that kind of wraps up our session for 2025.
Thanks, everyone. Hope you have a great rest of your day.
Thank you.
Thanks all.